This is the last meeting of the semester, right? I actually had that wrong with my night class. Just want to be sure. Next week is the official last week of classes for the semester, but Thursday of next week follows a Wednesday schedule, which is part of dealing with all the snow days that we had at the beginning of the semester. So this is our last class. We don't have class next week. The week after that is finals, when we'll be taking the second practical exam. I got started a little late, so I didn't do all the setup I would normally do before class started. So right now, the next week folder is actually pointing to the reproductive system, which is what we're doing this week, and I haven't changed it to point to the practical folder here. So on the 14th, which is 2 weeks from now, we'll be taking the second lab practical for the semester. The outline and the cases are in this folder for you to prepare with. There's the online assignment, and there's also a cumulative final assignment. To be sure, have I talked about the cumulative final with you guys yet? Okay, just want to be sure I didn't leave that out. The cumulative final and the practical are due. They have due dates for when you come in for that practical. You'll be taking that along with my Tuesday class. And there's not enough room in here for both classes to sit down at the same time. So I'm going to stagger the start times. The Tuesday class will start at 12:30, which is when they normally have their class on Tuesdays, but it'll be on Thursdays. They'll start at 12:30. You'll start at 1:30, okay, just to stagger it so there's enough seats for everybody. Even though you're starting an hour late as far as the official final exam period is stated, you will have the entire hour and 50 minutes that is allotted if you need it. So you'll start at 1:30. If you need to go till 3:20, that's fine. The period's officially over at 2:20, but because of getting a seat in the room, I'm staggering it. So you will have until the 3:20 if you need it, based on that later start time. There shouldn't be any conflict for you with that, unless for some strange reason you had a 3:45 Tuesday afternoon class. That's, I think, the next exam there. So I don't think it'll be a conflict for anybody, because you're already usually in here from 1 to 4 on Thursdays. But that's the plan there. So the due dates for the practical and the cumulative final that are in this folder officially say 12:30 on the 14th, but I actually won't shut off the access to Blackboard until you're physically in this room for that test. So if between 12:30 and 1:30 you want to finish a couple of last little things on Blackboard, you'll still be able to do that. Okay, I won't get in here and turn it off until everybody's here for the, but until 1:30 for your class start time. But keep in mind, that is the last chance you have to complete stuff. Everything has to be done before you come in here. You won't be able to get on Blackboard after you leave here from the practical to do any more work. That's the absolute last time to do anything on Blackboard. So, is everybody clear on that? Yes. Okay. Today's material, again, is on the reproductive system. I was actually a little confused with my Tuesday night class. I thought that they were, at that point in the semester, I was actually off by a week. But while I was eating dinner on Tuesday before that class, I got into my Prezi presentation on this material, and I started finally updating it with current pictures from the book, which is something I've been meaning to do for everything, but I've only gotten around to doing it for this one. So I'm actually going to use this presentation material a little bit more extensively. I've used this kind of material in the class before, and I usually have to say, this is a picture out of another book, that kind of thing. But now, most of the pictures in here are coming out of this book. Not all of them are, because there are some pictures that the book doesn't have a good representation of. And I'll explain that when we get to them. This splash page here, I just like this picture. It's a scanning electron micrograph of an egg cell covered in sperm, which would be what happens when the egg cell gets fertilized. But it's a great picture because it shows you the relative size of the two types of cells. So each one of these little things is a sperm cell with its flagellum coming off. And then you can see a suggestion of the spherical shape of the egg cell here, which is much larger. So I kind of like that depiction there. This picture is actually a little bit artificial, probably because it's prepared through an artificial insemination technique. But what's misleading about this picture is that there wouldn't really be that many sperm cells on the egg cell at the point of fertilization in the actual reproductive system. And if I get a chance to, I'll talk a little bit about why that is, but I don't always get around to that. So what I want to start with, or what? Actually, let me lay out the whole day for some before I get on. Usually what I do is, well, usually at this point in the semester, I'm kind of struggling to make sure I cover everything in my more normal lecture style class. And I don't have a whole lot of time for the reproductive system in lecture. So what I've tended to do over the years is to make the laboratory material about the male reproductive system and the lecture material about the female reproductive system. And mention a couple of things that kind of correspond between the two in either setting. But so what I want to do today is kind of in the first half address the male system, and in the second half address the female system. The book kind of lays it out that way anyway. So in the chapter on the reproductive system, the first section is anatomy and physiology of the male reproductive system, and the second section is female system. And then it has a section about development of the reproductive systems. I don't tend to deal too much with developmental topics within this. We usually just deal with the fully formed adult system. So really we're dealing with those two major parts of this chapter, male system and female. And the book kind of lays it out the way that I want to approach it. There is another chapter after this in the book, which is development and inheritance. It's not a chapter that you're going to have any chapter assignments from, but I do want to pull a little bit of information kind of out of that chapter, just to set up the reproductive system a little bit, which will sort of set the stage for everything we're going to talk about. So my expectation is to get through sort of what I do for lab in the first half for most of my classes, and then the second half will be more about the lecture material. There's a little bit more to be said about the female system and the male system time-wise, so I'll probably start into the female system before we take our break at the midpoint, but that's sort of the way it's going to break down. So first I want to talk about some issues just about reproduction, about what the system is meant for. And the picture I was just showing you there is an egg that's being fertilized. And then these pictures, which again are out of the next chapter about development and inheritance, but I just wanted to bring in some of those pictures because it shows what's going on. This is what happens after fertilization. So that fertilized egg cell divides into 2 cells. And then those 2 cells each divide, giving rise to 4 cells. And then those 4 cells each divide, giving rise to 8. And then 8 to 16, 16 to 32, 32 to 64, 128, 256, 512, 1024, 2098, 4096, et cetera, et cetera. Those are just the powers of 2. So 2 times 2, 4 times 2, 8 times 2, et cetera, et cetera. Just exponential growth there. And I've actually never stopped really counted, Venero. You know how many powers I just went through? Is that 8 or 9?
Oh, it was 10.
10?
2048, the game is a game called 2048.
Okay.
And you put 2 numbers together with 10 power.
That's 10, okay.
Yeah.
So 10 rounds of cell replication, starting with a single cell. I guess it's up to, sorry, 2000-ish? Is that what it was?
Yeah, 2097.
Whatever. It gets up to a whole bunch of cells. I'm actually not positive how long it really takes to get that far into making all the cells to make up the embryo. Here, the last couple of pictures there are the blastocyst stage, which it says is about 70 to 100 cells. So it's not that far through it, but that's probably about 4 or 5 days after fertilization. And the cells are starting to differentiate into different regions within this structure. There's the inner cell mass, which is going to become the embryo itself, and then the outer part, the trophoblast, is going to develop into structures having to do with the placenta and that sort of thing. Now, all of what's happening here is a single fertilized egg cell is just making copies of itself. The cell division that gives rise to all of these cells we're talking about is what's called mitosis, which is sort of a simple, straightforward type of cell division. Technically, mitosis is talking about what's happening in the nucleus, and it's followed by the full cell dividing. But we usually think of it as cell division. So in mitosis, what happens is the DNA in the nucleus duplicates, and then it segregates into 2 new nuclei, and then those 2 new nuclei are going to each be part of a resulting cell. So those 2 cells we see there have at their core a copy of the nucleus that was in the fertilized egg cell, and so on and so forth. All of these cells are basically duplicates of that original cell at the genetic level. In mitosis, we see clonal copying. Okay? Probably when I say the word clone, your first impression is going to be more of a Hollywood meaning of clone, which should be, an Arnold Schwarzenegger movie or something like that. But actually, when we talk about a clone in more formal science terms, it just means that the DNA is identical between 2 instances. And so here I'm talking about cells that are clones of each other. If we're talking about Dolly the sheep, who was the first cloned sheep, that sheep was made by taking the DNA out of another sheep and injecting it into the cell in place of into the egg cell in place of the fertilized nucleus. And so that new egg cell developed into a new sheep using the DNA from its progenitor. So Dolly is a clonal copy of another sheep because it has the exact same DNA. All of the cells that we see dividing here are clonal copies of each other. They have the exact same DNA. What makes them different, in fact, down in the blastocyst part of the picture, we're starting to see the cells are differentiating to different types of cells. What makes them different is what parts of the DNA they use. So all of the cells, with a few exceptions in your body, have the exact same DNA, but what makes a neuron different from a muscle cell that's different from an epidermal cell that's different from a fibroblast is just which genes in the DNA it's using. So a fibroblast is going to use the collagen gene to make collagen. An epidermal cell is going to use the keratin gene to make keratin. A muscle cell is going to use the myosin gene, and a neuron is going to use the voltage-gated sodium channel gene. Okay. It's not just that one gene. It's all of the genes that are necessary for a cell to be able to do that particular function. That's where they differentiate. Okay. But all of the cells go through the exact same type of division, this mitosis. But to get to the point where we have a fertilized egg cell, we actually have to introduce a different type of cell. So the egg and the sperm are special cells that are generated not through mitosis, but through a different process called meiosis, which is how we generate the sex cells, or the gametes, which are the egg and the sperm. And in that, instead of just making an identical copy of the progenitor cell, at least at the DNA level, in meiosis, what's produced are, from a single cell, 4 cells that each have half the amount of DNA as the original. Now, it's not just, DNA by weight, but this one has a gram of DNA and these 4 each have a half a gram, which is not at all a realistic weight for DNA in a cell. But it's specifically the pieces of DNA that we see in the nucleus of a cell. What do we call the individual DNA molecules that are in the nucleus of any cell?
Haploids?
Yeah, they're chromosomes.
Chromosomes.
Okay. How many chromosomes do you find in most of the cells in your body?
46.
46 chromosomes really defines the genome of humans. The gametes have 23 chromosomes each. Okay? And so when you put 2 gametes together that each have 23 chromosomes, you end up with 46. The 46 chromosomes you have are in pairs, 23 pairs. And in each pair, you have 1 chromosome that you got from your mom and 1 that you got from your dad. So the gametes are just mom's chromosomes in the eggs and dad's chromosomes in the sperm, and they came together to fertilize the egg cell and end up with the complete 46 chromosomes that we see there. Now, this picture is a representation of what we call a karyotype. And it's a look at the different chromosomes in a cell. Now, this picture was probably produced, well, actually it's probably produced by digital means, but it's based on sort of the old-fashioned way of doing this, where you take a picture of a cell that's going through mitotic division. And this, right before mitosis starts, all of the DNA in the nucleus duplicates to make a copy of itself and condenses into chromosomes. And then, so you take a picture of the nucleus and all the chromosomes have condensed down into these shapes that we can see here. And then the old-fashioned way of doing this with, paper photographs and that sort of thing is to actually take a pair of scissors and cut all the chromosomes out and then line them up so that they match. Now, the stain that's used on these cells highlights bands of patterns within the chromosomes, which help us to understand which chromosome we're looking at. So the chromosomes get paired together in this cut and paste technique. And we can tell that 2 chromosomes are the same because they share banding patterns. So for instance, these 2 next to each other, their banding patterns are pretty similar. They don't look identical because these aren't perfect images of the chromosomes, but we know that these 2 are a pair here. These 2 are a pair. There are 22 pairs, which are 2 homologous, nearly identical chromosomes. 1 from mom and 1 from dad in each pair. I haven't finished.
Oh, sorry.
Hold on. Those 22 chromosome pairs are ranked by number based on their length. So chromosome number 1 is the longest, and chromosome 22 technically is meant to be the shortest. 21 and 22 are very close together in size, and it actually turns out that 21 is a little bit shorter than 22, but before really good techniques of figuring out the length of DNA came along, somebody thought 22 was the shortest, and so it got numbered 22. And they're not going to switch it because the numbers now fit with the banding patterns we associate with chromosomes. There is, however, a 23rd pair. Those are the sex chromosomes. So in this karyotype, we see 2 chromosomes there at the end, which are not identical. And those are the X and Y chromosomes. Now, chromosomes really have kind of an X shape to them in this fashion. There are 2 strands, 2 chromatids that are attached in the center, and so they have kind of an X appearance. But the letter X was used to describe that chromosome just because somebody said, hey, it looks like an X. And then the other one's called the Y chromosome. Some sources will try to tell you that it looks like a Y. That's really kind of a little bit of a push, but it's really called the Y chromosome because Y obviously comes after X in the alphabet. When we're looking at this karyotype, since there's an X and a Y chromosome there, we know this karyotype comes from a male. Okay, men have an X and a Y chromosome. Women have 2 X chromosomes. So if we're looking at a female karyotype, we'd see 23 pairs of identical looking chromosomes. And since we're looking at a male karyotype, the sex chromosomes are not identical because of the Y chromosome. The presence of the Y chromosome causes the embryo to masculinize and develop into a male offspring. And the absence of the Y chromosome leads to sort of the default, which is feminization of the embryo. When this man develops sperm, these 46 chromosomes are going to end up being split between the gametes that are produced through meiosis. And so there's a 50-50 chance whether those sperm are going to carry a copy of his X chromosome or a copy of his Y chromosome. For a woman, she only has X chromosomes in that 23rd pair, so all of the eggs are going to have an X chromosome. When an X-carrying sperm fertilizes an egg cell, there'll be 2 X chromosomes, and therefore that's going to be female. When a Y-carrying sperm, Y chromosome-carrying sperm fertilizes an egg cell, since there's already an X in the egg and a Y comes along, that means it's going to lead to male offspring. There are a few other things that have an impact on gender determination, which I have some stories about that, but I don't think I'm going to have time to talk about. They are kind of interesting, but we have other things to get into. Anyways, so these are the chromosomes we see in any cell, and to put together the correct combination and number of chromosomes for the next human offspring, these specialized sex cells are responsible for doing that. Now, before I get into talking about the cell division in a little bit more detail and setting up talking about those sex cells, I do want to take a little bit of a tangent with chromosomes because I think it's kind of interesting, and it's does address a few reproductive functions that aren't often considered too much in a human A&P course like this. So this picture is the cover of a journal called the American Biology Teacher. And it's talking about an article which, well, there's an article in this magazine about it, but it's talking about an article that was published, I think, in Nature magazine, which is one of the really big biology or science journals, which was comparing the chromosomes of 4 different species. So they're not labeled here, and I can't entirely remember, but I definitely remember the first chromosome in each set of 4 is a human chromosome. And then the other 3 are 3 other great ape primates. I think it's a chimpanzee, a gorilla, and a bonobo. It might be orangutan instead of one of those others. Well, definitely one of them's a chimp, but gorilla, orangutan, bonobo. I don't remember which of those three are the other two. What they're doing is they're just comparing chromosomes across these different species. And I was saying earlier with the karyotype that you can figure out which two are chromosomes number 1 because they have similar banding patterns. These pictures, these chromosome maps, are showing those banding patterns in a lot of detail. We can line up these 4 species chromosomes next to each other and see how the banding patterns actually match quite a bit across all the chromosomes we see here, showing some homology between these species. But then also there's some variation. So while in this area of chromosomes number 1 for all of the species, there seems to be a lot of homology. For humans, there's something extra right here, right in here, which makes chromosome 1 a little longer, and its midpoint is offset from the others. But then from the midpoint up, the banding pattern that we see kind of does correspond. Okay, but since the other 3 are shorter, it looks like they're offset, and that's really because of this part offsetting. Or whatever species this is, it has a little extra something at the end or at the top up here that doesn't quite correspond with the other. So there's a little variation, but overall, across all the chromosomes we're looking at, there's a lot of similarities between all of these primates, which makes sense because we are genetically very similar species. One big difference that you see, however, is where it says chromosome number 2, that's really referring to the human chromosome number 2, which we see here. And these other species don't have a corresponding chromosome to that exactly. Instead, in each of the other species, there are 2 chromosomes, which we can match up the banding patterns and see that this chromosome is very similar to this side of human chromosome number 2, while this chromosome is very similar to this side, which suggests somewhere in the speciation between humans and these other great apes, these 2 chromosomes merged together in an ancestor that gave rise to humans. Now, whether that chromosomal change is exactly what made humans different from all the rest is hard to say because there are a lot of other things. But this is a pretty significant difference we can see right here. So, stupid presentation thing. Sorry, the presentation freezes up every so often. But so looking at all of these, they're numbered 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 22, and the sex chromosomes are these down here. For humans, those are the numbers like I just showed you in the karyotype. For the other species, they actually have 2 chromosomes corresponding to our chromosome number 2. The numbers are a little annoying here, but which is to say, while humans have 23 pairs of chromosomes, chimps and other organisms, these other species, have 24 pairs of chromosomes. Part of what got me kind of interested in this was a show I watched on, I think it was the Discovery Channel, at least a decade ago. It was a show called Humanity. And much like some of these kind of lesser cable channels today, you know, where you can watch Ancient Aliens and, you know, what is it, Ice Road Truckers and, you know, Catfish Hunting or whatever those shows are. They come up with these shows that are kind of very sensationalistic, trying to pass it off as, you know, educational programming. So Humanity, it's a 2-hour show, and for an hour and a half, they're talking about this particular animal that seemed to be a mix of human and chimpanzee. So they're calling it Humanity. They're suggesting, sort of obliquely, a lot of nudge, nudge, wink, wink, maybe it's a hybrid human chimpanzee. It did not look like your average chimpanzee, but it didn't look like human either. It stood upright, walked upright. It had a bald head, although it had fur over the rest of its body. And it just didn't quite look like a chimpanzee. and it had some, at least behavioral features that really looked human. And so they're going on and on about, you know, how this could be, and explain a little bit about hybrids, and make some rather off-color suggestions of how human sperm and chimpanzees' eggs might come into contact. And in fact, they're making a point that it would be human sperm coming from the man and chimpanzee eggs, because it's not likely for the opposite to happen. Anyway, so a lot of kind of suggestive things like this for about an hour and a half. And then the last half hour, they basically say, ha, we gotcha, it's not a hybrid. And the main reason why that's not possible is what we see here. Okay? Humans have 23 pairs of chromosomes. Chimpanzees have 24 pairs of chromosomes. One of the functions in the reproductive system that doesn't usually get addressed in human AMP is how it's very hard for different species to intermix. And it's nearly impossible for there to be a human chimpanzee hybrid, because a 23 chromosome carrying sperm and a 24 chromosome carrying egg cell, if they tried to fertilize, would not fertilize properly. The mismatch in chromosomes would say, hey, there's a problem there. Now, there are some situations like Down syndrome where there could be the wrong number of chromosomes. But in Down syndrome, what actually happens is chromosome number 21 doesn't segregate properly in the process of making an egg cell. So the egg cell ends up with 2 maternal chromosomes 21, and then the sperm cell brings along its own chromosome 21. So there's an extra chromosome there. But 21, like I said, is actually the shortest chromosome, aside from the Y chromosome. And so there's not really enough DNA in that chromosome to throw off the fertilization process. So that fertilization will be successful. It'll implant and come to term, but there will be problems because of the extra chromosome that leads to Down syndrome. But a big difference, like it's these chromosomes right here. So human chromosome 2, and then these chromosomes from the chimpanzee that are obviously the 2 halves that came together to make chromosome number 2 for humans at some point. If those 3 chromosomes came together during fertilization, they're so big, they're big pieces of DNA, that would really kind of throw a wrench in the works. It would never happen. So it's impossible for humans to hybridize with chimpanzees. However, it is possible for hybrids to happen in other species. And I have a picture here. I just really like, oh, stupid presentation. Keeps freezing up on me. I have this other picture here of a hybrid animal. This is called a liger. It is the offspring of a male lion and a female tiger. Its claim to fame mostly comes from the movie Napoleon Dynamite. For some reason, that character was obsessed with ligers. But what's interesting about ligers is that they're the biggest cat. And the reason for that is because the genes that regulate growth are found in the female lion genome and in the male tiger genome. So when a liger is produced, it does not get the growth regulating genes from either parent, because it gets the male lion genome and the female tiger genome. And for those 2 species, it's the other parent that would contribute the genes that regulates growth. So ligers just keep growing. This is an impressive picture, a really big freaking cat. But actually the woman standing there who apparently has some connection to ligers, I think it's down in Florida, there's a big cat preserve. So I've seen several pictures of her. Sometimes she's a brunette, but she's a fairly petite woman, which really kind of exaggerates the size of the liger. But there are other pictures of ligers. If you're interested, go ahead and do a Google image search. And there's one really nice one, which I wish I'd saved and put in here, of two handlers with a liger, and it's standing up on its hind paws, and it's like twice as tall as a human when it's standing up like that. So they're huge. Question?
Wait, so it's the female will be from the tiger, right?
So the name liger is the male is the lion and the female is the tiger.
So is it both the opposite around? It doesn't.
So there is a hybrid called a tigon, which is a male tiger and female lion.
Alright.
And they are actually rather small.
Oh.
Now, there's a little cub on the back of the liger in this picture. That is not a tigon.
Okay.
I don't think that's a tigon. That's a cub. Tigons are just small for those species. They're not like house cat small. They're just small for big cat species because they have sort of a double dose of the growth limiting genes in them. But so this is an example of hybrid, and I like this because it's kind of a neat little species. The thing about tigers, tigons and ligers is that they don't reproduce on their own. They're infertile hybrids, which is common in hybrids. So even if fertilization of the egg cell is successful and it implants and comes to term, and there's an organism that can survive and grow to full adult size, they aren't likely to be able to reproduce. The most common example of that is the mule. Mules are not fertile. You can't take two mules and get them together and make a new mule. If you want a new mule, you have to get a horse and a donkey and put them together. Now, I can't remember, but a mule is actually specifically a male of one species meeting with a female of the other. And then the reverse has a different name. I think it's called a hinny or something like that. But I don't think people usually make that big of a difference between the two. But again, they're infertile. So they are not viable organisms. They can't propagate a new species or anything like that, or hybrids. There are a number of different kinds of hybrids that are out there. I was trying to find some good pictures of hybrids, and doing a Google search for hybrid images, images of hybrid organisms, is not very successful thanks to Photoshop. Most of the pictures are just these weird chimeras that people have put together in Photoshop. But I did, though, in doing the searches, both for the liger and the tigon, both searches came up with a picture of the parents making the offspring. So searching for liger, there was a picture of a male lion mounting a female tiger. Looking for tigon, there was a picture of a male tiger mounting a female lion, which I decided not to put in here, but it does kind of paint the picture for you. Anyways, so those are hybrids. And I really just went off on this tangent to kind of reinforce the idea of how the reproductive system has built into it some functions that perpetuate the species specifically. There are no known, and I doubt there are even any possible, human hybrids outside of science fiction, because our genome is really distinct from any organisms that are similar enough for something like that to possibly happen. So these big cats can hybridize because there are a lot of things about them that make it possible for them to mate. But if humans having 46 chromosomes found an organism with 46 chromosomes, if they're not physiologically compatible, then they're not going to hybridize. I don't remember, I did search a while back for other organisms with 46 chromosomes. And I think the only one I could find was some plant species. So it's certainly not possible for a human and a plant to hybridize. And so there are a lot of problems trying to bring, sperm and a seed together and make them into a plant-human hybrid or something like that. But
Are people with Down syndrome infertile?
I'm actually not positive. I get a no back
I
here.
don't believe so. I've seen shows where they've had like Down syndrome couples having kids.
Okay, so.
I think there was a documentary actually.
Actually, I'm not surprised that they're not infertile, because when they make gametes, half of those gametes are going to be normal. So let me get to making gametes thing, and I'll come back to that idea. So I sort of started this off talking about cell division. And so I have two pictures here. These are not pictures from the book. These are pictures from Wiki Commons, which is a repository for non-copyrighted images. But I went back, well, there's nothing about this in the chapters in Unit 6. And I went back to the cell chapter in Unit 1 to see if there are pictures that I could use from the book there. And I don't like the pictures they have explaining mitosis. And they don't have any pictures explaining meiosis. So I did a search and I found this. And I actually kind of like this. I've never seen it presented quite this way. This picture and the meiosis picture I'm going to show you in a second are made by the same person. They have kind of a common theme to them. And one is mitosis is a cycle. This is how cells reproduce over and over again. So here's a cell. It goes through some changes, and I don't really care that you learn the names of the different phases, but it grows and then it copies its DNA into the more X-shaped chromosome that I talked about before. So this red X-shaped chromosome here is really just this red line, which is a chromosome that's duplicated itself. And then in what we call really mitosis, the chromosomes organize along the center of the nucleus on these spindle fibers, and then half of each duplicated chromosome separates off to make two identical nuclei that are the same as the nucleus here before the duplication started. And then the cell divides into two. And so talking about this being a cycle, they have one cell going through the cycle again, and the other cell splits off from that. Now, if this is a growing tissue, there might be both of these cells go into another cycle of reproduction, or in more like maintenance situations, say in the stratum basale of your epidermis, one cell continues on and makes and divides again, and the other cell ends up differentiating into the mature, in that case, keratinocyte. But the point here is that after going through mitosis, we end up with two identical cells, clonal copies of each other. And meiosis is a little bit different. First off, one of the points that the artist here is trying to make is meiosis is not a cycle. So we don't have a closed loop here. Instead, we're starting with a cell here and ending with 4 different types of cells at the end. Meiosis has 2 rounds of division. So here we are with a cell kind of like what was at the beginning of where I talked about mitosis a second ago. It has chromosomes represented by just a single line. The DNA there duplicates and we get the X shape like we saw before. And then once they go into meiosis, the first round of meiosis, which is the first of 2 divisions, has the chromosomes lining up along the center, but in paired groups. That's kind of redundant. In pairs. So this long red chromosome here and this long blue chromosome here would be the maternal and paternal chromosomes number 1, let's say. So they both duplicate. So we now have a duplicated copy of the maternal chromosome 1 and a duplicated copy of the paternal chromosome 1. They then line up next to each other in meiosis, and a little bit of the DNA can cross over, swap between one chromosome and the next. So as we go into this point and beyond, there's a little bit of blue on the red chromosome here and a little bit of red on the blue chromosome here because it's swapped, physically swapped a little bit of DNA back and forth. That increases the genetic variation that we have in the chromosomes. Then it continues on through division, kind of similar to what happened in mitosis, except that instead of separating the duplicated chromosomes, one of each chromosome from a pair goes to one side and one goes to the other side. So now we have two nuclei that each have two chromosomes instead of the 4 chromosomes originally. The chromosomes are duplicated, they're still in that X shape, but there's only 2 of them in each nucleus. So we've cut the number of chromosomes down in half. Okay, it's still duplicated DNA, but they're cut down in half. And then each of these cells, after they divide, go through another round of division. There's no new DNA duplication. Okay? So these X-shaped chromosomes now in the second round separate half of the chromosome to either side. So we now get back to the single line chromosomes like we started here. It's just that we have 4 cells that came from that one that each have half the number of chromosomes. And also in this picture, that swapping where a little bit of red mixed with the blue and a little bit of blue mixed with the red shows us that we have in these 4 unique sets of chromosomes. Okay, so here we have one that's a long red and a short blue. Here we have one that's a long red mixed with some blue and a short blue mixed with some red. Here we have long blue mixed with some red and short red. And here we have long blue with nothing mixed and short red mixed with a little blue. Okay. So all 4 of those are unique in what DNA actually ends up in them, but they have half the number of chromosomes as the original. If one of these meets up with the gamete from the other gender to fertilize, then we have a new cell that has the original 4 chromosomes, like the cell that started all this out. That's meiosis. In mitosis and meiosis, there are names of the different phases going on. I don't care that you know those. Being in A&P, I'm assuming that you've had some biology background, so you went into learning the phases of mitosis and meiosis there. We're not going to get into that. Rather, I just need you to be familiar with what's happening and the idea that we start with one cell with pairs of chromosomes and we end with 4 cells of single chromosomes. Okay, so that the 2 cells with single chromosomes can come together and reform a full genome of another human being. And in each of these, they have 2 arrows, one pointing from the cell and one pointing from nothing, coming together that says zygote. Okay, so 2 of these cells can't combine to make a zygote, which is going to become an embryo. It has to combine with another gamete from the other gender. So this is just showing that these 4 cells would all want to combine with another cell to make zygotes. It's possible. Now, this is very general. This meiosis isn't specifically talking about sperm or egg cells. We'll see a little bit more specifically what's happening for the 2, because once we get to this stage between sperm and egg, what happens is quite different. But generally speaking, this is the idea. 1 cell gives rise to 4. I should say, when we're talking about these cells that have pairs of chromosomes or single chromosomes, we use some particular language, which actually is in this picture. But I want to introduce that language real quick. The terms haploid and diploid describe the chromosome numbers that we see in these different cells. Haploid means 1 each of, how do I want to say this? 1 of each type of chromosome. Okay, so chromosome. So 1 chromosome 1, 1 chromosome 2, et cetera, et cetera. Diploid means pairs of each type of chromosome. Each type of chromosome. So 2 chromosomes number 1, 2 chromosomes number 2, et cetera, et cetera. Okay. The prefix in diploid means 2, so it's 2 of each chromosome. The prefix in haploid kind of means 1 or whole or unity or something like that. But a lot of students will look at that word and imagine that it sounds like half, which is not correct as far as what the word really means, but it can be very helpful. Diploid is twice as many chromosomes as haploid. Haploid's half as many chromosomes as diploid. It's very circular to define them that way, and it's technically incorrect. But if it helps you remember which is which, that's fine.
Why don't you just call it monoploid? Oh, uniploid. Why don't you just put like mono or uni?
You're welcome to go back in time and tell the people that came up with these words. You have to learn how to speak ancient Greek before you do that, though.
Yeah.
Or actually, would that be Greek or? I don't know. It's Greek or Latin, one of the two. Haploid cells are gametes, and diploid cells are everything else, body cells. Okay. Basically put. For animals, it's pretty simple. That's all they are. Plants are kind of confusing in their reproductive cycles, and I definitely don't want to get into that. But so actually, yeah, for animals, it might be say, why don't we call it uni and di? But when we get to plants, there's a reason probably why that word's used instead. You will sometimes see, and in the book they use this, gametes or haploid cells are denoted by N, and body cells or diploid cells are denoted by 2N. N just is the number of unique chromosomes, which is 23 for humans. So gametes have 23, and other body cells have 46, just 2 times 23. In humans, denoting it as N and 2N isn't really necessary because we know what that N stands for. But if we're talking about just reproductive systems in general, the number varies from species to species. Like in chimpanzees, N is 24 instead of 23. If I remember correctly, there's a species of fern that its N number or ploidy number is 234. So it has a lot of chromosomes. It's either 234 or that's its diploid number. I can't remember, but the N is just a variable depending on the species, and of course they're only talking about one species here. But I wanted to point that out because we will see that in the book. And in fact, in the book, there's one kind of weird place where they put one N instead of just N. They actually do it both ways, and I'll point that out. It'll be pretty obvious. But so I just wanted to define those terms. In the picture I was just showing you, they don't actually use those terms to denote things. Instead, they have C and N, for some reason, I don't quite know why, is referring to, I guess, the long and the short lines here. So there's 2 long and 2 short. And then when they're duplicated, there's 4. Oh, I see what they're doing. So C here stands for chromosome, and N is the ploidy number. And so for this organism, N is 2. So the diploid cell has 4 chromosomes. And then here where it says 2C times 2, that's meaning that the chromosomes are doubling as the DNA is being duplicated. That's what that's saying there. Anyways, so those are the 2 different types of cell division. And I'm taking a lot of time here at the beginning to set this up because the point of the reproductive system that we're going to focus on is really the production and delivery of these gametes. And so that's what the male reproductive system is about, is making sperm and delivering them. And then the female reproductive system is making egg cells. And then the 2 types of gametes combined in the female reproductive system. And then it's the rest of it's about gestation and that sort of thing. But we're going to concentrate on where the gametes are made and how they move through the reproductive tracts of the two genders. Okay. So we want to start with the male reproductive system. This picture is showing kind of all of the components of the male reproductive tract. And I just want to run through it real quick with this picture, and then I'm going to go back and talk about different components of it in a little bit more depth. So sperm are made in the male reproductive gland called the testis, which is this egg-shaped gland right here, which is kind of unfortunate because it's not making eggs, it's making sperm, but they decided to color it white. So that's where the sperm are made, and then they move into this structure called the epididymis, and the sperm migrates through the epididymis and then onto this tube that leaves the scrotum where the testis is found and gets into the pelvic region. And that tube is not labeled right here. The label is actually way up here. That's the ductus deferens. You've probably heard reference to it as the vas deferens, but vas deferens actually isn't the correct term for it. Vas means vessel, like a blood vessel or a lymphatic vessel. And by definition, vessels have a simple squamous epithelium. Ductus deferens is referring to the fact that this is actually a duct, which means that it's a tube that does not have a simple squamous epithelium. The epithelium lining the male reproductive tract for most of the distance is a pseudostratified epithelium. And we'll see some micrographs in a little bit, and I'll point that out to you. You've probably heard of a procedure called a vasectomy, which means to cut out part of the vas deferens. So by removing a little bit of the tube right here where it emerges out of the scrotum, it interrupts the male reproductive tract, and so sperm can't get up there, and that is a form of birth control. So the vas deferens, ductus deferens continues. It comes out of the scrotum, goes around the pelvic bone, gets into the pelvic cavity. It goes alongside the bladder here, and then there's this little thicker area here called the ampulla of the ductus deferens, which is where the ductus deferens ends. And it's right alongside this structure called the seminal vesicle. Those two come together right here, right at the edge of the prostate gland. The tube that is common coming out of both of those is called the ejaculatory duct, which passes through the prostate gland and joins up with the urethra. Now, the urethra, which we talked about last week, is part of the urinary system. And so this part of the urethra right here is only urinary tract urethra. But right here, where it meets up with the ejaculatory duct, from this point on, the urethra is both urinary and reproductive tract structure. The urethra emerges from the prostate. Just beyond the prostate, we see the muscles which contain the external urethral sphincter, and then continues along the length of the penis. Right here at the base of the penis, another tube's coming in, and this is the duct of the bulbourethral gland, which secretes contents into the urethra. The seminal vesicle, the prostate gland, and the bulbourethral gland all secrete fluids that feed into the male reproductive tract. The seminal vesicle and the prostate gland are mostly making secretions or fluids that have to do with what the sperm is suspended within. Sperm need a source of energy to power mitochondria that make the flagellum move. So they need a source of carbohydrates. So those are present. Urine is a little bit acidic, so some of the secretion is going to be basic to neutralize the pH in the urethra. And the lining of the vagina is also acidic to be antimicrobial, so it has to help buffer that too. So those three glands all play a part in that. The prostate and seminal vesicle are mostly about the seminal fluid or the ejaculate fluid. And then the bulbourethral gland secretes some fluid pre-ejaculatory to help prepare the urethra for the passage of sperm through. And that's really where the buffers to neutralize the environment of the urethra come from. So, and then the sperm leave through the external urethral opening to be introduced to the female reproductive tract. So that's the whole thing. I just want to go back and talk about a few things in more detail with some pictures. First off, the testes are suspended in the scrotum, which is outside the pelvis. So here's a few pictures of structures related to the pelvis. What's kind of most important to mention here are the muscles in the middle one, the cremaster and dartos muscles, which play a role in elevating the scrotum. The usual story for why testes are found in the scrotum is about temperature control. Spermatogenesis, the meiotic divisions that lead to making sperm, happen best at a slightly low temperature. So body temperature is 37 degrees Celsius. Spermatogenesis really should be taking place about 34 degrees Celsius. So usually people will talk about this scrotum being there to help regulate the temperature in the testes. When it's cold, these muscles will contract and pull the testes closer to the body to keep them warm with body heat. And when it's warm, these muscles relax and the testes suspend away from the body where they can get more air to help keep them cool. That's generally the explanation. I was reading an article about 2 years ago now, challenging that usual story. Not all, well, we'll just stick with mammals. Not all mammals have external testes. And so by looking at the different solutions to this problem, it suggests that that temperature control thing isn't really the point. Although they add to that, but probably not the original reason for the scrotum. Because those animals that don't have a scrotum, that have internalized testes, still have to deal with the temperature thing. And they deal with it by helping radiating off heat around the testes and keeping them cool relative to the surrounding body temperature. So it is possible to regulate the temperature of the testes within the body. Okay, other species do that. This study I was reading was talking about what they found is extrapelvic testes correspond mostly to organisms that run. So their sort of go-to example of an internalized testis animal was a dolphin. And dolphins, of course, don't have a scrotum, which would be sort of counterproductive to their streamlining so they can swim long. But there are other organisms they mentioned, I can't remember all of them, that have internalized testes. But when you move to organisms like a gazelle or predatory animals or their prey that run quite a bit, that's when you start to see these externalized testes. And the suggestion from the study was it had to do with blood supply. The movement around the hip joint required for running can potentially damage the blood vessels going to the testes. So organisms that run very fast externalize their testes so that the blood supply is removed from where the hip joint is. So that's the suggestion. I just mentioned this because the temperature regulation is a very prevalent story on why they're there, and you might come across that, but just understand that those kinds of explanations that don't have really good data supporting them are usually just somebody said, hey, I bet this is why. But it's probably explained in a different way. And so this other study that I'm talking about actually did look at different species and what made them different and why they had scrotum or not. So. Looking inside the scrotum, we can see the testis, the epididymis, and the beginning of the ductus deferens here. This testis is really just a big mass of tubes. The first person to ever successfully perform a dissection on a testis, and back in that time when you performed a dissection, it was a theater event. Not like going to the movies now, but scientists would get together at medical schools and an anatomist would do a dissection in front of everybody, and that's how they learn about what was inside the body. So there's actually a story about the guy that first did this successfully with the testis. And at that time, not really understanding how reproduction worked, people thought inside the testes were little itty bitty humanoids that traveled along the reproductive tract and got into the female system and grew large there, and that the woman didn't really have anything to do other than gestation. They didn't understand gametes and that sort of thing. So they expected inside there'd be something completely different. And what they expected, I'm not entirely sure. But this anatomist was doing a dissection, and he just very gingerly cut along the outer wall, the tunica albuginea. That's probably not how you pronounce it. And all of these tubes just spilled out. And I was just like, oh my God, what's that? It's like spaghetti falling out of a Ziploc bag or something. And nobody was expecting that. I mean, the guy that did it was, because he'd done it before. But when it was first done in public, everybody was like, oh my God, I didn't know that's what was in there. They're really just a bunch of tubes. And inside these tubes, in the wall of these tubes, is where sperm are made. And then they travel along these tubes and leave the testis to get into the epididymis, which is just a bunch more tubes. And then those tubes condense down into a single pathway, and at the tail of it, epididymis, they turn around and start to make up the beginning of the ductus deferens here. And then we see a single tube clearly here, extending away and then going up out of the scrotum. The spermatic cord, which is labeled at the top there, is basically the connective and muscle tissues and blood vessels and that sort of thing that surround the ductus deferens. And it extends out and into the pelvic cavity through what's called the inguinal canal there. This is a micrograph of a cross-section of one of those tubes in the testis. Okay, so this is the epithelial wall there. Now, I said a little bit ago that the epithelium of the male reproductive tract is a pseudostratified epithelium. And in fact, that's what we're looking at here. Although it doesn't really look like it. It doesn't look like the pseudostratified epithelium of the trachea, because there's also meiotic cell division going on here. So a lot of the nuclei that we see in here are actually nuclei of cells that are dividing that are becoming sperm. And then there are some that are the actual epithelial cells, which are the Sertoli cells here, what the book also calls sustentacular cells. So they're pointing at a cell, I guess, right there, which is part of the actual epithelium. And then a lot of the other cells here are ones that are going through meiotic division. Now, this is a picture summarizing the meiotic divisions that eventually produce sperm. There's a diploid cell designated by 2N called a spermatogonium that's in this layer of tissue. It undergoes a mitotic division, making 2 clonal copies of itself. One of those just remains as a spermatogonium as the stem cell for making these. And the other one is called a primary spermatocyte. It goes through it goes through the first meiotic division here, giving rise to the secondary spermatocytes. This is a place in the book where they're kind of weird. It's 2N and 1N here instead of 2N and just plain old N, like I was showing before. But these cells, the secondary spermatocytes, are haploid cells. Okay? They still have the duplicated chromosomes, but they only have one of each chromosome. And so they divide, and half of each of those single duplicated chromosomes end up in the gametes at the end here, which are spermatids, again designated 1N instead of N. After this happens, these 4 cells then become spermatozoa, or sperm cells. And spermiogenesis is the process of the kind of boring old spermatid cells getting rid of a lot of their excess cytoplasm and growing flagella. In the sperm cell, really all we have is a nucleus and a motorboat. Okay? So all of the cytoplasm, or not all of it, but most all of the cytoplasm is sloughed off, and we just have the nucleus left over with that haploid set of chromosomes. It's covered by, or surrounded by, a layer called the acrosome, which is going to be important for helping it interact with the egg cell. What is left of the cytoplasm is just what's wrapped around the base of the flagellum here. And there's a whole bunch of mitochondria there. And those mitochondria are going to be making ATP like crazy to help power the movement of the flagellum. And then the flagellum would just be, what makes it swim. This is a highly specialized type of cell that develops at the end after these kind of generic looking cells here come out of the 2 rounds of meiotic division. And then they specialize to do this, which is different from how it works with eggs, but that's how the sperm come to be. And so in here, what's happening throughout the whole thickness of the wall of the tube are these cells going through these divisions. And then right at the edge are going to be the cells that go through spermiogenesis. So this is pointing out early spermatids. And then the actual spermatozoa are going to be right at the edge, if you could even see them. And usually what you can see are a bunch of flagella kind of sticking out into the lumen here, but that's not really obvious here. Probably because this tissue, as it was prepared, a lot of the sperm kind of fell off. But there's that. Now, the book does not have a picture showing you any more detail about the seminal vesicles or the ampulla of the ductus deferens or the ejaculatory duct and the prostate gland. This is a picture that I pulled off the Wikipedia Commons, which is a Gray's Anatomy drawing. So sort of a defining book in anatomy was Gray's Anatomy, not the show starring Meredith and Dreamy. But Dr. Gray drew a lot of pictures that are sort of the definitive anatomy things. And they're all in public domain because it's a very old book. And so they're all in Wikipedia. So I just pulled this up because it's a copyright-free image to put in here. But just to show you, this is on the posterior surface of the bladder. And the vas deferens, or the ductus deferens, has come in, up and around the bladder. And it gets a little thicker right here as the ampulla, where the lumen starts to get a little bit more tortuous. And it meets up with the seminal vesicle, which we see opened up here. And so these are ducts coming out of the seminal vesicle where the ejaculate fluid is going to mix with the sperm as they're traveling along here. And that happens when they go into the ejaculatory duct. The ejaculatory duct is just a short little duct that connects to the urethra right here. And there's going to be one on either side because up to this point, everything's bilateral. But then from the urethra on, there's just the one tube going along length there. So I just grabbed this picture just to give you a little bit of an idea of what these structures look like. It kind of looks like an apple in this drawing to me, sort of like hanging off an apple tree or whatever. But, then this picture is showing the structures that have to do with the length of the penis and how an erection is achieved. So in the picture of the penis that we see here, the sagittal views, the urethra is surrounded by this stippled-looking spongy tissue, and then along the top, there are these little bits and pieces that kind of the same appearance. All of that's the erectile tissue. That tissue has a blood supply that when blood gets to the tissue, all of those blood vessels dilate and the tissue becomes engorged with blood, and that's what makes the erectile tissue erect. What's surrounding the urethra looks really big in the sagittal view. Okay? That's what's called the corpus spongiosum. But in this cross section, it's actually a fairly small component. And why it's drawn this way in the pictures, I'm never quite sure, because it doesn't look like quite that big. And then what looks rather small in the sagittal section is what's called the corpora cavernosa, which are the two columns of erectile tissue that run along the dorsal surface of the shaft of the penis. Corpora cavernosa is Latin pluralization. A single one of these would be called a corpus cavernosum, just like corpus spongiosum. But we usually refer to them as a pair, corpora cavernosa. So at the top, this is the flaccid view, so there's not much blood in the erectile tissue. And in the erect version, that spongiosum and the corpora cavernosa are all engorged with blood, and that's what leads to erection. Okay. I'm going to point this out here, but I'm actually going to come back to it when we talk about the female system. This is how the hypothalamus and the pituitary gland regulate sperm production. There's a hormone released from the hypothalamus, which is abbreviated GnH. Yeah, okay. So where it says number 1 up there at the top, they have it abbreviated twice properly, GnRH. But then next to the red arrow, they got the H and the R backwards. Oh. So it's GnRH, which is gonadotropin releasing hormone. And that's just a hypothalamic hormone that regulates anterior pituitary function. And it causes the release of gonadotropins. The gonadotropins are FSH and LH. In terms of the male reproductive system, it's best to just refer to those by their abbreviations, because what FSH and LH stand for is really about their function in the female system. So I'll talk about them in more depth with the female system. I'll come back to this picture later on to remind you of this and connect the hormonal function here with the hormonal function in the female system. But what these hormones do, FSH stimulates the cells of the seminiferous tubules, which is where the sperm are being made, the tubule we were just looking at, to promote sperm generation, spermatogenesis. Then LH stimulates cells in the interstitial space, the connective tissue around the tubes, called Leydig cells or interstitial cells, to produce testosterone. Now, in this picture, they're really just talking about the regulation parts of things. So testosterone feeds back and inhibits gonadotropin release and gonadotropin releasing hormone release up here. So this is just a negative feedback signal telling the regulatory centers that the Leydig cells have gotten the signal and they're making testosterone, leave them alone already. And also, the Sertoli cells make inhibin, which obviously from its name is an inhibitory signal. It's also going to limit FSH and LH release there. What's going on in the female system is very similar to this, but just a little bit more complex. So after talking about the female system, we'll come back to this. I'll remind you about how they're kind of similar there. Which brings us to the female system. So we'll go ahead and take a break now. I was actually kind of thinking I'd get through the first stuff a little bit quicker and we'd kind of start this, but we'll go ahead and take a break now. And then we'll come back for a nice romp through female reproductive function. And I say that because I'm going to be talking fast to make sure I cover everything before Wednesday. But so I'll be back in here by 1:45 by that clock. The sooner you're back, the sooner I get to start, and the slower I can talk to finish up the day. But 20 minutes is our usual time. So, but if everybody's back sooner, we'll get started again soon.
Professors, there are questions.